Um, in the previous session, if you were here a few weeks ago, I told you that today we were going to discuss this particular paper, T-cell circuits that sense antigen density with an ultrasensitive threshold. So what I like to do today is to tell you more about this idea of how you can use synthetic biology, and hopefully after today's talk, all these cartoons are going to make sense. I won't give you a lot of details right now, but what I like you to point out is that the tool that I'm going to describe today should allow T cells to now do a specific tumor recognition. And this will be really helpful for sparing normal tissues and targeting only cancer cells using this technology that is chimeric antigen receptor T cells. So I'm going to start by introducing very briefly, what's the fundamentals behind this approach in cancer immunotherapy. And then I'm going to dive into the figures of the manuscript. No, so the paper I'm telling you about came out a few years ago, and we are still continue working on a second layer of this technology in my lab. But so just I encourage you to send questions as we go through the slides, and if something is not clear, just I'll be trying to monitor not the chat, but also feel free to just unmute yourself and just ask a question. So just to give you a brief introduction, again, the idea is that now we can engineer immune cells to kill cancer. And what I'm showing you here is a microscopy trajectory of T cells that are labeled in yellow and tumor cells that are in green. And one very fundamental concept about the new technologies of chimeric antigen receptor is that we are using the fundamental properties of these cells, such as the navigation through the human body and the very complicated identification processes that the T cells can perform, to redirect them and to make them kill other cells. No, so T cells also have the ability already to defend us against virus, bacteria, even some forms of cancer, and we are using those abilities, the ability to proliferate and to kill, but now we can redirect them against cancer. It says screen is not showing the microscopy. You cannot see the movies?
No, I can't see the movie.
Don't you see like some things like floating around and green things on the screen?
No, we are still on the first slide.
Oh, what's going on? Okay, let me see. It says your screen sharing is paused. Okay, how do I? Hey, sorry about that. Something happened.
No worries.
I don't know what exactly. Okay, how about now?
Yeah,
Why
I see.
does it say your screen share is paused? I don't understand. Do you see, for example, if I move to the next to this one, no, you are not seeing my full animation. Do I need to click something in particular?
I don't think so. Are you trying to move on to the next screen?
Yes, resume share. I'm seeing resume share. Yeah, because you can see maybe my movie now, but if I move to the next, Do you see the transition? I don't know if you are seeing the transitions.
No, I don't see the transition.
Yeah.
Maybe we play the movie, and then once the movie is done, and then we can reshare the screen again to move on to a slide. Do you think that would work?
I don't know if it's a issue because right now, for example, I'm moving to other slides and I don't think you are seeing that either,
Okay,
right?
yeah.
So I'm right now I'm moving towards different. Sorry.
Someone Jasmine suggested maybe it's easier to share the entire screen zero rather than just a specific
Okay,
tab.
let me see. Let me see if sharing my full desktop, but I don't know if that will help. This is the first time I have any issues like this.
Yeah, I haven't seen it before.
So do you see
Yeah,
my slides?
yes,
Is it advancing now?
yeah, yes, yes.
Okay, so then I will just move on in this one. So if you can see this movie, then the idea is that T cells can be engineered to recognize these proteins that are on the surface of cancer cells. And in that way, you can redirect the cytotoxic activity of T cells against cancer cells. So this approach is called chimeric antigen receptor T cells because we are using these synthetic receptors that are kind of like a Frankenstein between so these receptors have a recognition domain on the surface of these constructs, and then in the intracellular domains of these receptors, you have parts that are coming from the native T cell receptor or other costimulatory proteins, so that all together, when these T cells engage with a target, then they start killing. But the challenge is that these receptors, when you put them into the engineered T cells, they will kill any cell that they encounter. These T cells with these receptors, they will kill any cell that they encounter, regardless of the amount of antigen that is expressed on the surface of the target cells. And this is a problem because, for example, these antigens or proteins like HER2 and EGFR and GD2, so those are proteins that are highly, highly abundant on the surface of tumor cells, but they are also present at lower levels in the surface of other normal tissues. So you could imagine, no, if you try to engineer these T cells against any of these proteins, they are very potent, they are very powerful, like these T cells, but they could result in problems of toxicity. So, and to give you just an idea of what will happen, for example, I'm going to show you in a moment a movie where you see again these microscopy images of cells that express low levels of this antigen HER2 in green to your left slightly. And then on to the right of that, so there is this other co-culture with cells that express high levels of the antigen in red. And then the T cells are engineered against this protein HER2, and they are depicted in blue. So if you run this co-cultures assay and take pictures every 2 hours, and what I'm going to show you is actually is sped up so that it only takes a few seconds. What you will see is that both cell types, the green cells and the red cells, which are different in the amount of this protein HER2 that is expressed on their surface, both of them are getting killed. So this is not good because that could result in problems of toxicity. No, if you were to put these engineered T cells back into patient, most likely this will result in problems of toxicity. And in fact, for the protein HER2 and also several other proteins, there has been reports of this toxicity of what people call on target, because these T cells are recognizing the intended protein, but of tumor toxicity, no? So because it's not being recognized in the tumor, but it's being recognized maybe in other tissues. So what we wanted to do, and which is the purpose of today's talk, is to engineer a new behavior into so that we can only kill the cells that express the high levels of the antigen, but spare cells that express low levels. And this behavior, this sigmoidal behavior, is called ultrasensitive or sigmoidal, no? And what I'm gonna do today is to introduce you to the tools that we use to engineer this behavior. So I need to tell you a little bit about this synthetic notch receptor. No, notch is a receptor that is involved in cell-cell contacts. And for example, the extracellular portion of notch here in yellow binds to its ligands, one of them being delta. And when this binding event happens, there is a cleavage event of the intracellular portion of this receptor. So these are proteins that are sitting on the membrane of cells, and the intracellular part of this receptor is a transcription factor, meaning it can traffic to the nucleus and then start activation of any downstream program. So what Leo Marsud and Cole Roybal, who were a few years ago postdocs in the lab of Wendell Lim, now they are professors at UCSF and USC. What they thought about was what if we keep this functionality of notch, but now use it in a synthetic manner to encode novel functions, no? And what they did was to replace the outside part of this receptor for any recognition domain of your choice. For example, you can use the binding domain of an antibody. And in the intracellular part, you can replace this endogenous transcription factor for a synthetic transcription factor. So that now the way to think about this synthetic notch receptor is that they convert binding into transcription activation. And so then you can convert any binding of your choice into the expression of any payload that you choose as well. No, all of this through methods that I'm gonna tell you a little bit more in a few slides. But throughout the years, what is been shown is that these synthetic notch receptors are very helpful to encode a variety of functions across different cell types. For example, if you are interested in identifying contacts between different cell types, you can use synthetic notch receptor. If you want to start a downstream program, for example, differentiation or multicellular patterning, you can use this synthetic notch receptor, and you could also use these receptors to encode more complex logic functions. No, for example, AND gates, OR gates, no, and so on and so forth. So the way I use this synthetic notch receptor is shown again in this diagram, where what we thought about was to control the expression of the chimeric antigen receptor. So I told you earlier that these chimeric antigen receptors are the ones responsible for activating the T cells for killing. But rather than having those receptors always on the surface of the T cells, what we did was to put it under the control of the synthetic notch receptor. So the synthetic notch receptor is shown here to your left. And the idea is that it's gonna recognize the same antigen than the CAR, but it's gonna do it with a binder that has low affinity for that protein. And it will control the expression of the CAR in a second step. So the way this synthetic notch receptor should work is that if it encounters cells that express low levels of the antigen, because the affinity of this binder is low and because the abundance of the protein is low, then the frequency of interaction between the protein and the receptor should be low, which then should avoid the CAR expression. In contrast, if the same cells now encounter tumor cells that express high levels of these proteins, that should increase the frequency of interaction between the synthetic notch receptor and the target protein. And that should lead then to the activation of the CAR. And because the CAR has high affinity, so meaning it will want to stick to the protein, so then that should lock these T cells to engage with these target cells and start the activation of killing and proliferation. So what I want to do in the next slides is to tell you about what experiments we did to test this design. No, but let me quickly check. Yeah, if there is any question at this point, because this is one of the most important slides. No, it seems that there is no question, so I will just continue to hopefully so far we are on the same page. Okay. So the first thing that we wanted to do to test this circuit design was to build a system that the only difference among the target cells was the antigen expression. So, and we used, or we picked this protein HER2. I mentioned earlier, HER2 is a protein that is highly abundant on the surface of cancer cells, for example, some subtypes of breast cancer, ovarian cancer, gastric cancer, but it's also present on the surface of a lot of tissues, for example, the skin or heart, lungs. And but we wanted to, because there is difference potentially on the growth rate of these cells, the media that they use to grow in the lab. So there is many variables. So we wanted to build a system that was highly controlled and very well calibrated. So we built a series of cancer cells or cells that express different levels of this protein HER2. And we measure what those levels are. So for example, the high levels are in 10 to the 7, no? It's like this is 10 million molecules of HER2 per cell. Whereas something that has kind of lower levels or closer to normal tissue will be something in the order of 10 to the 4.5 or so, no? Which is like 10,000 molecules per cell. So the normal tissues and cancer cells differ, at least in the context of HER2, in roughly 100-fold difference in expression. So we made sure that the engineered cell lines were roughly resembling the levels that you will find in cancer, and similarly, the cells that express the low levels that resemble the low levels that you will find in normal tissue. So our system was comparable to other studies that have been done by other groups. For example, here all the way to your left, you have the expression of an ovarian cancer cell line. And then here to your right, across all different tissues, is the levels of HER2 expression across a variety of tissues. No, for example, neurons express HER2, or cardiac myocytes, skeletal muscle. So there is several cells that express HER2, but at lower levels. So I told you that you need binders of different affinities. So we started from the scFv of this molecule called Herceptin, and we introduced point mutations to generate a series of recognition domains that have different affinities. So starting from something that is roughly 2 nanomolar to something that is lower affinity. And so in this case, the numbers are opposite, no? Like a smaller number means higher affinity, 2 nanomolar, and then, for example, 200 nanomolar. So you can then start making these co-cultures. No, I showed you earlier that you can combine cancer cells and T cells and use microscopy or other methods like flow cytometry, where you can count accurately how many cells remain alive after, no, for example, 3 days of being together, the T cells and cancer cells. And so you can quantify in that way what is the percentage of cell killing or cell lysis. And here what I'm showing you is the result of some of these assays, where in the x-axis you have the antigen density of the HER2, and on the y-axis you have the target cell killing, and on your left there is the results for these circuits, no, the SynNotch-CAR, and on the right you have the results for T cells that all the time express on its surface the CAR. And hopefully you can see that in the context of the circuits, you get these sigmoidal responses that we were looking for. And in the context of the T cells that express all the time these chimeric receptors, then there is no discrimination. So there is like this kind of more like um, um, all regardless of the antigen expression, you are getting T cell killing, no, of the targets. So in addition to these results, you could do measurements of what is the CAR expression on those T cells that were co-cultured with targets that have different expression. No? So these are plots that are coming from a technique called flow cytometry, where these are distributions. So you cannot see here, but like these histograms are coming from individual cells. So you can aggregate all that data and plot this into a histogram, and say for each of the cells, what is the CAR expression. So to quantify the CAR, so this again, the molecule in green, we attach to it a fluorescent protein, so that using flow cytometry, we can measure how much of this CAR has been expressed when you have T cells that express, that are co-cultured with targets that have different expression levels. And again, hopefully you can see that the larger the density, no, so these are cells that express 10 to the 6.9 molecules of HER2 per cell. So the larger the amount of the CAR on the surface of these T cells. So that's a very important aspect of how the system works. And the other one is the T cell proliferation. So the way these experiments work is that you label the T cells with a dye, and every time that the T cells divide, then the dye is gonna have to dilute between the two daughter cells. So the larger the number of divisions, the amount of fluorescence of that dye is going to get diluted on a per cell basis. No? So, for example, if you start with T cells that are not dividing, then you will get this population here at the bottom, no, and signal that is maybe 10 to the 5.1. But you see how as I go up on these plots, there are a larger number of peaks, and then cells that also express now or that have lower amounts of signal. No, for example, here at the top there is some cells that now have 10 to the 4.5 or 4.231, and so that means that the cells divided more. So this is very important because the way the T cell or this circuit works is has two main components. One is the component of the controlling of the CAR expression. That is, again, the receptor that is going to activate the T cells for star killing. And the other component is the component of the T cell proliferation. No? So now not only you are going to have cells that are expressing the CAR, but also you are going to have more cells. No? So the T cells are going to start dividing, and then that results overall in this sigmoidal behavior. So I'm going to show you now what happens if you do similar microscopy experiments as the one I showed you earlier, no? Which previously I showed you that T cells that express all the time the CAR molecule will kill both, no, cancer cells and cells that express lower levels of the antigen. And now I'm showing you the microscopy effect, microscopy trajectories of T cells that were engineered with this circuit, no synthetic notch low affinity to chimeric antigen receptor high affinity. And you can see that only the cells that express the high levels of the antigen are the ones that are getting killed. Whereas on your right, you see how the cells here in green are completely covering the surface of these plates and were not damaged. The other thing that maybe you can see from these microscopy images is the concept that I just alluded to one slide ago, which is that you see a lot more blue dots, no, in the cells that were co-cultured with the target cells that express high levels of the antigen, and fewer dots. So that even though both started with the same amount of T cells, at the end, you end up with more T cells in the co-cultures with the high HER2 targets. No, so that's the concept of controlling the CAR, but also controlling the proliferation. Another set of assays that we did, as I showed you earlier, these target cells were plated on a flat surface to help with the microscopy. But you could also imagine doing these 3D spheroid models to try to mimic a little bit better the spatial geometry of the cancer cells and the solid tumors. So in these assays that I'm going to show you in a moment, what we did was to build two different cell lines, one that express low levels and another that express high levels of HER2. And we use a cell line that is known to be very good at forming 3D spheroids or three-dimensional tumoroids. No, so these are something that assembles into small spheres. So we can use also a dye that will allow us to measure the cells that are dead. And this dye is called caspase dye. So we can build these spheroids and culture in 3D. And what I'm showing you here is just a projection of these spheroids, but these are kind of little blobs. In red, you see the spheroids that are expressing low levels, and in blue, the ones that express the high levels. And in orange and in these yellow dots are the T cells. So if you co-culture these spheroids with T cells that do not have the receptor, nothing happens. No, like both spheroids are alive. And here I'm showing you representative images, but I'll show you in a moment histograms where we quantified hundreds of these spheroids, not to have the statistics. If you have now spheroids of both low HER2 and high HER2, but in the presence of T cells that express all the time the CAR, then you see both types of spheroids getting destroyed. And in the context of spheroids that are co-cultured with the circuit T cells, the SynNotch-CAR, only the spheroids with the high HER2 are getting killed. The others are, as you can see here, are still round and intact, and we don't detect the green signal. So you can like measure that, as I said, for hundreds or even thousands of these spheroids. No, so here the numbers at the bottom tell you how many of these spheroids we quantified, and then plot the distribution of the green signal on a per spheroid basis. And so hopefully you can see that again, the T cells that express the CAR all the time on the surface kill both, no, the high and low spheroids. The circuit only kill the high HER2 spheroids, and the low HER2 spheroids are being intact or at the same level that you will find when you use T cells that do not have these receptors. The additional assays that you can do with this type of spheroids are shown here, no? Where you mix both spheroids that have low levels of HER2 and spheroids that have high levels, with the idea that you can quantify what will be the potential of target killing. No, you could imagine these T cells could get activated into the tumor and then from there traveling to other spheroids to start killing them. So one way to start assessing that question was to do these mixed spheroid cultures. So what you will see here is how the caspase reporter, which is again the marker for T cell killing, it's primarily found where the high HER2 spheroids are. So there is very little of target killing. So again, you could use the same approach of quantifying on a per spheroid basis and see how the majority of the signal is concentrated at the center, no, because that's where the high density spheroids are. And then as you move away from these high HER2 spheroids, then the signal starts decaying. Okay, I'm gonna jump ahead and I showed you this before. So this is just the quantitation of the movies that I showed you earlier, no, how the T cells only kill the high HER2. So I'm gonna show you a last portion of experiments and then I'm gonna leave some time so that you can ask questions. So, but one next layer of complexity in these experiments is to have a mouse model, no? And in this context, or what we did here was to implant two tumors into the same animal. One tumor has high levels of the antigen, and then the other tumor has low levels of the antigen. And after a few days, we injected these T cells that have this circuit, low affinity SynNotch to high affinity CAR. And then you can measure over time the size of these tumors. And that is what is shown here at the bottom left. No, this is the days after tumor inoculation. And then on the y-axis is the volume of the tumor. So you can see how if in these black and gray lines, if you don't have any receptor that targets the HER2 antigen, you see how these both tumors grow. But in the context of the high and low HER2 density tumors, the high HER2 one was growing, but then around day 15, that stops growing and then now it shrinks. In contrast, the other tumor that expresses low levels of the antigen it continues growing. And then if you try to repeat this experiment and then around day 20 or so, you resect the tumors and then look for the T cells and count how many T cells you will find. So then you will, what we found is that you find many more T cells in the high density tumors. And also, if you measure what is the CAR expression on those T cells, just like before, what I showed you earlier in the context of the in vitro assays, you will find that these T cells that were pulled or that were removed from the high HER2 tumor also have higher levels of CAR expression. You can repeat similar assays, but now for different types of tumors, for example, ovarian cancer or breast cancer, no, that express 10 to the 7 molecules of HER2. and use others as proxies for normal tissue, something that expresses very low levels. And then you can see that only the tumors that express the high levels are getting killed. And one last piece of information, you could do also engineering of the T cells to have these luminescence reporters. So these are T cells that are being engineered so that when you add into these animals the luciferin, which is the substrate for the luciferase, which is the enzyme, then that will make the T cells glow. You will find that the T cells are mostly localized within the high HER2 tumors. No? So again, this is consistent with the in vitro data that I show you, where the T cells primarily will find the cells that express the high levels of HER2 and then proliferate within that context. Okay, just to summarize, what I show you today is how we are using synthetic biology to encode new functions. And today I told you this strategy to engineer antigen density sensitivity, meaning now the T cells have the ability to recognize not only the presence of a protein or a tumor-associated antigen, but also the amount of the protein and decide whether to kill or not those cells based on that amount. So in the context of T cells that encounter cells that have low antigen expression, then there is no CAR expression and therefore not killing. And if these T cells encounter cells that express high levels of the antigen, then that results in robust CAR activation and proliferation that overall gives you this sigmoidal response. And the tunable antigen density discrimination is a powerful tool to continue using an approach like engineered T cells for the context of cancer. So I'm just going to briefly tell you 1 more minute what are some limitations of these studies, and then I will open up the floor, I guess, for discussion or questions. One of the big limitations of this study is that at the moment, the animal experiments do not take into account the presence of the endogenous immune system. So what we're using is these animal models that are immunocompromised. And the reason for that is because that's the only way in which we can implant human cancer cell lines into a mouse host. Now, if you were to try to implant a human cancer cell into fully immune competent mouse model, just like what will happen in humans, no, like the immune system will try to reject that, no, and it will start killing those cancer cells, which will confound or will complicate our analysis, not to know whether the killing effect or behavior is coming from our engineered T cells or is coming from the endogenous immune system. And the other important limitation is that we cannot truly assess what's the effect of the tissue distribution of the antigen. No, like again, we use this low density tumor as a proxy for normal tissues, but we are only confining to that other tumor the presence of the low antigen expression, because the human HER2 and mouse HER2 are different enough that these recognition domains, these binders, are not cross-reactive. So one that is, so these proteins that we are using to recognize the HER2 protein, the variant for human HER2 is not recognizing the mouse HER2. So follow-up experiments and things that we have been doing and we are doing in my lab right now is to um, adapt the circuit strategy to fully immune competent model. And also now we are engineering mouse T cells, and we are using a transgenic mice where the tissues where before there was mouse HER2 expression, now they have human HER2 expression, so that we can do this experiment like or that more closely resemble the clinical setting, no, where only the tumors have the high density of expression of HER2, and then there is several other tissues that express the same protein at lower levels. Okay, so with that, I wanna again mention you my lab. No, so I don't have the all the names of my collaborators when I was at UCSF here, but again, just to recap, this was work done when I was a postdoc in the lab of Wendell Lim at UCSF, and it was a collaboration with the lab of Seth Gardner and Kalle Saxila at University of Finland. So this is my current lab. And so I'm going to stop sharing and I will take some of your questions. Okay. So there is one question here from Nishat. Um, I understand high HER2 tumors are the main focus of this formula, but we would like to know your opinion on if the low density tumors will communicate with the tumor microenvironment, considering high and low HER2 cells are found in the same region, and then adapt to avert the action of CAR T cells. In other words, could this cause the low HER2 to develop some degree of resistance to the CAR T cells? Yeah, so I will try to rephrase the question, but this is a very important point, and thank you for the question. So in the context of HER2 and some subsets of breast cancer, for example, um, one important aspect is the tumor antigen heterogeneity, meaning in the tumor, maybe not all the cells are going to express the same levels of HER2. So there might be variabilities of the levels, and maybe some of them are high and some others could be intermediate or lower and still are tumor cells. And that's a very important point, but because what we want is a therapy that is able to deal with this antigen density heterogeneity, but only confined to the tumor microenvironment. No, so we don't want the T cells to then traffic to other normal tissues and have this of tumor toxicity. So that's another aspect, a very important point that we are exploring currently in my lab. Not trying to design therapies that are going to be able to deal with different levels of antigen density, but without being toxic to other tissues. So that's a good point, and that's something that we are studying at the moment. There is another comment or question here from Jasmine. Is there a way to make this tissue specific to avoid on target of tumor results in normal cells that have high expression in HER2? Perhaps putting the circuit behind a tissue specific promoter or enhancer. Also, how would you modulate this system to reduce the specificity between non-cancerous cells with endogenously high levels of HER2 expression, such as in processes with neurogenesis, synaptic plasticity in the brain, heart muscle growth? Yes, tissue repair in response to injury. Yeah. So the question or the question slash suggestion is, are there other strategies to do this tissue specificity activation? Um, or how do we deal, no, in the case of if there are cells that we know that are healthy but express the high levels? Yeah, how do we avoid those cells being targeted? And that's a very good point. So as suggested, you could add additional signals or layer additional programs into the behavior of these T cells. One of them could be tissue specificity. For example, so that you say only if it's the pancreas or the colon, no? So then activate this circuit. So you could actually layer uh, more than one synthetic notch circuit to another synthetic notch circuit. That has not been done yet in the context of an actual therapeutic circuit, but there is a paper actually from a few years ago where there is these examples of layering, no, one circuit after another circuit, no, and in that manuscript, the antigens that were used were kind of dummy antigens, no? Like, for example, GFP or C19 or things that maybe altogether at the moment didn't make like a clinical sense, but that exemplify or show the capabilities of layering these circuits. You could also think about, for example, for sparing specific cells that we know are healthy, but you could try to do not gates, no, or other type of higher order logic gating, no, and SynNotch in combination with other tools that the field of T-cell engineering has created could be a way to also provide this higher order computation or logic into the T cell so that you spare other cells that are important. What generation of chimeric antigen receptor was used? So in our case, we use second generation. So these are T cells that have a transmembrane from CD8 and then 4-1BB costimulatory domain and CD3 zeta. Are there other questions? We still have like maybe 5 minutes or so. There's a question about considering higher generation cars in the future. Yeah, there is several um, approaches that you could use, not to layer this circuit or combine it with others. No, so we know that the tumor microenvironment and remodeling of the tumor microenvironment is important. So potentially combining this with cytokine secretion to remodel tumor microenvironment or other strategies could be used. So at the moment, not for the context of HER2 positive breast cancer, but in other contexts, we are using these synthetic circuits that, in addition to the CAR, also secrete cytokines, no, and that will be considered a third generation or a higher generation CARs, no. That's we often try to start from the simplest to the more complex, and the reason also because the more engineering you do, the harder it gets not to have enough cells to do assays. Also, introducing all these genetic circuits into the T cells, no, becomes more complicated as you start introducing more complexity. Does this circuit retain the functionality to undergo clonal expansion to extend to other regions, sites that have high HER2 expression throughout the body? Um, so that's a good question. I don't think we have done assays to try to get at that, but with the new model that we have, the immune competent system, that could be something to look into, no? For example, you could imagine what if you have primary tumor but also metastatic cancer, no? Uh, is this strategy gonna be good enough. We are also trying to quantify very accurately what is the off rate, no, the activation of these T cells. No, once they are in one tumor, how long does it take for those T cells to getting deactivated, move into other tissues, and either down regulate the CAR expression and then restart no their overall function, or whether they stay on, no, and there is a potential for of tumor toxicity. So those are questions that we are actively pursuing, and that we are trying to engineer as well. Okay, last question. The SynNotch-CAR T cell circuit demonstrate impressive specificity in discrimination between high and low antigen density cells. How might the delay activation of the circuit impact its efficacy in fast-growing or aggressive tumors? And what strategies could be employed to address this limitation? Yeah, that's a good question. As you alluded, and as I show you, there is some delay on the killing rate because it has to go through transcription and protein production, no, to start activating. But the activation rate is in the order of 12 hours or so, no, 12 to 16 hours, um, which we don't think that it's gonna be a big problem, no? Like even for fast-growing aggressive tumors, it's on the scale of weeks and months, no? And the activation rate that we are measuring is in the order of hours, no? But we are still exploring other ways to try to speed up the process, no? So you could imagine using stronger activators, no, to try to uh, um, speed up that process, or you could completely try to redesign a circuit with the tools that there is now on cell engineering. Might be possible to do circuits at the level of proteins. No, you could imagine maybe having a CAR or trapped, maybe already expressed but trapped in ER, and then upon a particular signal, release it, no, from so that the timing of activation maybe gets reduced to only then minutes or hours, no, but yeah, that could be a different type of approach if that were the case, no. But at the moment, we don't think that that is a major limitation. It's lower, but it's not. The activation rate is not that slower that will prevent um, efficacy. Okay, yeah. So thank you again, everybody, for joining today, and yeah, I hope this was helpful, and um, yeah, keep up the good work.